Let's get started on our second morning session. So our first speaker for this session is going to be Dr. Kerry Ressler. He's an HHI investigator and professor of psychiatry and behavioral sciences at Emory University. Dr. Ressler is a former MIT undergraduate and did his MD PhD work over at Harvard Medical School. So welcome back to Boston. And he's going to be telling us today about his work on epigenetic regulation of brain development and function. Dr. Ressler.
Thanks so much, Troy and Leeway. Hi, thanks. It's such a pleasure to be back, and MIT continues to just grow in such wonderful and bountiful ways, and it's wonderful to have this kind of symposium going on here where we can really start to explore how we can use the quantitative modern-day tools to address such difficult questions. So I'm going to be talking about data that really was wonderfully teed up by the two previous speakers. That's really spanning between humans with high histories of trauma exposure, and childhood trauma, and how we can understand the differential risk. One of the main questions that the first speaker brought up was why, even in these extraordinarily neglectful, and there's plenty of room still up front and in the middle if people want to sit, why, if you have these extraordinary neglectful and traumatizing backgrounds, still only 30% of the people develop really significant phenotypes and trauma sequelae. What is the difference? What is the individual variability? And we think part of the answer to individual variability for risk versus resilience to trauma exposure is genetics and epigenetics. So the first part of my talk, I'm going to be laying the groundwork for that, showing you a few examples in the system that's already been nicely talked about by the speakers in the HPA axis and CRH systems. And then the last part of my talk, I'm going to talk about some new data looking at intergenerational risk, where we have lots of anecdotal stories over the decades that offspring of those with a high level of trauma are at higher risk themselves for trauma-related sequelae. But it's been difficult to find animal model systems where we can really dissect that, and I want to share with you some new data that we're really excited about in that area. So the human data I'm going to be talking about is from Atlanta. Over the last 10 years, we've interviewed almost 8,000 people from inner city Atlanta in an epidemiological cross-sampling of people attending Grady Memorial Hospital, which is one of the largest remaining charity hospitals in the country. And we're interviewing people in all sorts of medical clinics, OBGYN clinics. But as Grady primarily serves the impoverished, the majority of the people we interview are very impoverished and very traumatized. So of these 8,000 people, it's great majority are minority populations, about 90% African Americans. Essentially everybody's under the poverty line. About half know somebody personally who's been murdered, about 2/3 have been attacked, and about 1/3 have been sexually assaulted. And the rates of childhood trauma, and so these are all adults, and we're doing now a study of intergenerational transmission with traumatized moms and their offspring, but I'm only going to talk about the adults and self-report data so far today. But of these adults, about 38% have had some level of significant adverse child experience or childhood maltreatment, with about 16% of them having at least one type of significant physical, sexual, or emotional abuse by age 12, and 22% having more than one. And it turns out that if you grow up in a difficult environment, multiple traumas is really the rule and not the exception. We have lots of buttons to press up here. Hopefully I'll be pressing the right one. So the focus that we have is what is the neural mechanisms of exposure to trauma at the level of adults in post-traumatic stress disorder and depression, at the level of kids in developing these systems. We've been exposed already to some of the main players, and I'll just restate. We think of the amygdala as the main hub of output of the fear response system, and I'll walk through more of that in a moment, being regulated constantly by hippocampal and prefrontal cortical areas that regulate emotional responding, as Kay nicely showed. Our focus is primarily on the amygdala, not because it's the only part of the story, but because it's a relatively simple and extremely robust part of the story. And if we take pretty much anybody in this room or around the world and we expose them in an fMRI magnet to a fearful versus a neutral face, you'll get activation of the amygdala bilaterally in an fMRI. And Amit Etkin and others have shown over and over again through many studies and meta-analyses that fear-related disorders, whether we're talking post-traumatic stress disorder or anxiety disorders like social anxiety, social phobia, or specific phobia, are all notable by their hyperactivation of the amygdala in these kinds of tasks. So what does it mean when your amygdala is activated? And I think the best way to get a sense of this clinically is the concept of a panic attack. So someone who's had a panic attack will say something like, all of a sudden I felt dizzy, my legs gave out on me, I couldn't catch my breath, it felt like someone was choking me, could feel my heart was beating too fast, terrified I was dying, I had to get away. And panic attacks are associated with cardiovascular changes, with somatosensory changes, with GI distress, with shortness of breath, difficulty breathing, sweating, lightheadedness, and really what I think we can think of as cognitive interpretations of all these expressions with sense of fear. Oops, I pressed the wrong one. And we can really think of a panic attack as a fear attack, I think, is a simplistic way of talking about it. And though we most associate panic attack with the term panic disorder, panic attack really occurs in all of these fear-related anxiety disorders clinically. So the difference is, what are the triggers? So with panic disorder, the panic attack seems to come out of the blue. You know, doc, I was fine, and then the panic attack came on, I go to the emergency department, think I'm having a heart attack, and in fact, it was a panic attack. But in fact, most people with panic disorder, if you take their history carefully and find the first time they had a panic attack, it often was at a time of high stress or trauma or depression or after something really horrible had happened. But rather than associating cognitively that panic attack with those bad traumas, they associate it with the interoceptive cues of their body, the heart racing, the sweating. So that the next time that happens, they're like, oh my God, it's going to happen again. And they start misappropriating the cues of the panic attack with the context around them, the people around them have to get away, and that's the idea of agoraphobia, where you start to avoid people and places because you're afraid you're going to have another panic attack. So it's really a generalization or a misappropriation of those cues. Simple phobia, if you're afraid of spiders and a big spider lands on you, you have all of those symptoms, we just call it being scared to death. But it's the same reflexive response. And similarly with PTSD, with the triggers that remind you of the trauma, the index trauma, activate those symptoms. And the reason I think this is such an important point is that decades of work by people like Mike Davis and Mike Fanzelo and Joe Ledoux and others have worked out the hardwired neural circuitry of amygdala output. And I think it's really the lowest hanging fruit in psychiatry for understanding how a complex circuit leads to a complex set of behaviors that we call a panic or fear attack. And we know now that hardwired central amygdala output through the hypothalamus in the brainstem leads to the heart rate and blood pressure changes, the vagus, the bradycardia, and GI distress, and other brainstem areas lead to all these symptoms we talked about. In particular, the paraventricular nucleus of the hypothalamus leads to corticosteroid release, and central to the brainstem also activates the dopamine VTA and the locus coeruleus systems that you heard about from Kay Tai. So I think in many of these cases where we're talking about panic or fear response, the question is no longer what's causing that behavior. We know what's causing that behavior at a neural circuit. The interesting question is, what's upstream of this? How is this being modulated? Why is one person having a fear response that's over-responsive and overactive, and other people are able to dampen it down? How are the amygdala being modulated by the hippocampus and prefrontal cortex in ways that lead to a resilient or normal responding versus pathological responding? So our focus is really how, given these children in foster care, how given people who live in high-risk inner-city populations, why does one person go on, or maybe on average about 2/3 go on to recover and have resiliency, but the other go on to have pathology, PTSD, depression? And we think we can break this down at multiple levels. First of all, at a genetic level, we know that the heritability of risk for PTSD and depression is about 30 or 40%. So that at the outset, people have about 30 or 40% of the risk, given trauma, to go down the high-risk pathway is genetically mediated. But what are those genes? We don't think it's one or two. We think it's hundreds. It's going to be hundreds or thousands. And they're going to all be intersecting at different points in this across this pathway. And we also know the environment's critically important. That's the point of today, that early developmental stress is one of the highest risk factors for all psychiatric disorders. We know that how people deal at the time of the trauma is critically important, that memories aren't created immediately, rather they're consolidated over minutes to hours. And one of our hopes eventually is by understanding the molecular mechanisms of consolidation, that one could have the same kinds of protocols in emergency departments that we have for stroke or for heart attack, where you could say, give this drug or do this therapy within the hours after the trauma and prevent the long-term sequelae of trauma and PTSD development. Finally, once it has become permanent, people go down different pathways, whether they're sensitizing or generalizing their responses or discriminating or extinguishing their responses seems to differentiate these two. And one of the main findings that has been really exciting in the field is the idea that extinction, as defined by Pavlov 100 years ago, diminished response to cues over time, is really the underlying component of cognitive behavioral exposure therapy. And that's really among the most empirically derived and consistent treatments for anxiety and trauma-related disorders. And now that we're understanding the neurobiology of extinction, it's leading to a whole host of new ways of thinking about how we can enhance the recovery process. But for the purposes of today, I'm going to talk about two sets of stories, which all have to do with pre-existing sensitivity. And the first is building on the pathways that we've already heard about with the hypothalamic pituitary adrenal axis, two genes in this pathway, and how those are regulated at a genetic and epigenetic way, and some hypotheses about how that may build upon the risk for later responding to trauma. And then secondly, this concept of intergenerational trauma, how the trauma histories of your parents may in fact affect the history and risk in this current generation. So again, our population is inner city Atlanta, and one of the main goals that we have is to capture a large enough database to eventually be able to do large-scale genetics on a hypothesis unbiased way to understand the gene pathways associated with risk. It used to be that 8,000 was a big sample. Nowadays, from a genetic perspective, it's a small sample. And what we know is that to do really large-scale unbiased genetics, you need on the order of 50 to 100,000. And this has been led really by the work of the schizophrenia field. Patrick Sullivan has led the Psychiatric Genomic Consortium. And what they've shown over about the last 4 or 5 years, that if they do whole genome association studies, so you're looking at about 1 million different places or polymorphisms throughout all the chromosomes, and then you're saying, what is the risk, the ratio of, this is a minus log p, but basically have to be over this line to be considered statistically significant across the entire genome. That once they've gotten over about 80,000 total subjects in about 20,000 cases, they've had enormous power to identify gene pathways that we really think is the underlying genomic architecture of schizophrenia. And it's really transforming the field, and you'll hear more from Danny Weinbergers and others over the course of the morning, how we can think about understanding these pathways. And PTSD is late to the game, but it's extraordinarily exciting where we are. In the last 2 years since we started the PTSD subgroup, we already have signed up about 30 different investigators from around the world with about 60,000 subjects, and that'll be about 10,000 to 15,000 cases. So though we're late to the game, we hope that what we're going to find, and that we've been talking about and will continue to talk about, is that we have a neural circuitry to overlay this genomic architecture on so that we can start to understand it and manipulate it relatively quickly. So while we're waiting for those samples to arrive and for the analyses to be done, we've been focused on a more targeted set of pathways, and that's our friends, the hypothalamic pituitary adrenal axis. So over the last about 8 years, we did a number of what we called targeted tag snipping approaches, where we took all the gene polymorphisms within about 10 or 20 genes associated with stress reactivity and started looking at how are those involved in these pathways for all the reasons that you've heard today. And just to remind you, the amygdala releases corticotropin-releasing hormone into the hypothalamus. CRH releases to the pituitary, which causes ACTH, or adrenocorticotropin-releasing hormone, release to the adrenal gland, which activates cortisol, which activates many of the stress fight or flight responses, but also feeds back onto the system to turn it back down. And we heard a lot about that with the diurnal variation of stress response by earlier this morning. So those two genes I'm going to talk to you about are CRHR1, which is the receptor for CRH. So it's one of the top level receptors in the HP axis field. And then secondarily, cortisol acts on a protein called the glucocorticoid receptor, GR, in its feedback. And GR is very highly modulated by a chaperone called FKBP5. And FKBP5 is turning out to be one of the most exciting proteins in the stress axis field because it seems to be the real master regulator of glucocorticoid receptor sensitivity versus resistance. And I'll show you some new data on how we're thinking about that. So this again is our samples are much larger than this now. This is several years old, but what we found early on was that we could replicate in our cohort the same findings everybody else is. More child abuse is really bad. so multiple quantitative levels of physical, sexual, emotional, childhood abuse and neglect. More abuse and neglect leads to more depression as an adult, or is correlated with more depression as an adult. But what's interesting, this is if you take the population as a whole, and it was referred to the idea of individual variability. Take-home message is if we subdivide this group up by polymorphisms within that CRHR1 receptor, that top-level receptor, we find this very interesting pattern. That this is across about 5 different polymorphisms that are all what we call in linkage disequilibrium. So they're different SNPs, but they all kind of travel together in the genome. And over again, what we find, and now this has been replicated by multiple groups around the world, is that one version of the gene is associated with much greater depression levels than another version of the gene that seems to not be associated with really any greater depression levels, despite the same level of moderate to severe child abuse. So that it's really, this differential genetic risk is associated with this individual variability for how people respond to this lifetime trauma. How can we examine these pathways? So Kei-Tai talked about some of the models that one can use, and our main focus is on fear conditioning for a lot of the reasons I told you earlier. We don't think fear is a model for PTSD, but we know people with PTSD have differential levels of fear, and people with depression have differential levels of avoidance responding. And so it's a really clean, robust model of understanding amygdala activity. And just to give you a very simple idea of how we do this is a classic picture from Joe Ledoux. We will take a mouse, habituate him in a context, do Pavlovian conditioning, pairing a tone or a sound or a light, or in the final studies I'll show you an odor, a smell, with a foot shock, and then you bring them back the next day in a different context and you play the cue. And you ask, are they more, if they've been paired to be afraid, they freeze a lot more than if they were not, did not have associative learning. And what we know, again, is that the central amygdala, which is the hardwired output of this fear pathway, is modulated by inputs from the lateral and basolateral amygdala, where the cues, both the unconditioned foot shock pain cues, are paired with the conditioned cues from the auditory system, the visual system, the olfactory system. And the plasticity that we'll hear more about here, and that's so much of the focus of the Picower, is happening right here where this emotional learning is occurring. And that's been being modulated by these prefrontal and hippocampal areas we talked about. So one set of studies, we've done a lot, and the field has done a lot in understanding the role of CRH, and so asking the question of, CRH polymorphisms are associated with fearful responding, with depression and PTSD-like responding. Can we understand that at a causal level? If we manipulate the CRH neurons and thus the CRH release, can we affect in a mouse model this process? And this has been done in lots of ways, and I'll just show you one very brief example of how you can do this. So in this case, with Liz Martin and Charlie Nemiroff and Mike Owens, we made a transgenic mouse in which the driver of the CRH receptor was driving Cre recombinase. And you heard about Cre from Kei-Tai. It allows you to cause recombination specifically in those cells. And what was nice is we could show in a reporter mouse that we only got expression in the central amygdala, the BNST, and the PVN very nicely. And that we could show co-expression of the levels we expected in the CRF neurons. So in one set of studies by Georgia Gafford in the lab, she specifically crossed these animals to a GABAergic receptor. So GABA is one of the main inhibitory receptors in the brain, and it's what causes cells to sort of dampen down. And the hypothesis is if you knock out the inhibitory receptor, the cells will be more active. So we think it's a mouse model where we've overactivated the endogenous CRH system by only activating GABA. And this shows that it worked. And the take-home message is this. If you knock out CRH GABA, so you overactivate the endogenous CRH system, the animals, once they're trained to be afraid, they stay afraid. They don't extinguish. And remember, extinction is part of that natural recovery process. They activate the HPA axis more rapidly with more corticosterone. They're more anxious. They spend less time in an open arm of a plus maze, and they spend less time in the open field of an open field maze. And finally, if you give a drug that blocks the CRH receptor, that gene that's associated with depression, you then rescue these anxiety and depression-like phenotypes of less time in the open arm and less time in the center. So you can do a lot of things like this, suggesting that there's a causal component there and really start to work out the circuitry and the phenomenology of that system. And furthermore, the CRH system's been associated both with polymorphisms of stress as well as corticosteroid responses to plasticity in the brain, and by Michael Meany's work that was referred to earlier in terms of this idea of intergenerational transmission or parental transmission of epigenetics. And I'm not going to talk about epigenetics of the CRH system, but I'm going to talk about the other one in just a second. So the other side of the HPA axis is glucocorticoid regulation. So that was at the top CRH. I'm going to go to the bottom and look at cortisol feedback. And one of the most replicated findings in post-traumatic stress disorder is hypersensitivity of the HPA axis. And one way of measuring this is what we call the dexamethasone suppression test. So this has been done for 30 years and replicated over and over in lots of different studies. Basically, if you take people's baseline level of cortisol, you give the cortisol agonist called dexamethasone the night before, and then you measure it again the next morning. Everybody suppresses some, but if you have PTSD, you suppress a lot more. You have hypersuppression or supersensitivity of the HP axis. So how is this regulated? FKBP5 turns out, as I suggested, to be the master regulator. So it's acting as a chaperone in the cytoplasm bound to a complex of glucocorticoid receptors. Normally, GR lives in the cytoplasm, and when cortisol binds, it translocates to the nucleus. And in that translocation, it's then able to act as a transcription factor and activate lots of downstream genes. Normally, FKBP5 is preventing that translocation, and when enough cortisol binds, it's displaced by FKBP4. Well, the cool thing is, when one of the main downstream transcriptional activators of GR is more FKBP5 itself, which you can then see acts as an intracellular negative regulator. And the take-home message is that polymorphisms in this gene that act on how GR activates the gene lead to different levels of FKBP5 responding, and thus alter whole body levels of GR sensitivity versus GR suppression, and regulate this HPA response. And I'll show you a little bit about that. So remember the graph I showed you a minute ago with the CRH polymorphisms. The same story. We looked at lots of polymorphisms in the FKBP5 gene, and several of those, shown by this one as one example, have the same similar process. But now it's not depression that we're seeing a difference on, it's PTSD. And what we find is that one version of the gene is associated with much more levels of PTSD compared to the opposite version of the gene. And if you just take the homozygous alleles, you see again the risk allele versus the resilience risk allele versus the resilience allele in the no abuse versus abuse category. So if you have a history of abuse, you have an enormous difference between these groups. And if you don't have a history of abuse, you have much less. And that's been shown, we've showed that across multiple different polymorphisms. What's interesting, and I'll come back to in a minute, is both the CRH polymorphisms and the FKBP5 polymorphisms are associated with childhood trauma, increasing risk for adult depression and adult PTSD, but do not associate with adult stress or adult trauma, increasing this. And Kay Tai mentioned a difference that she saw with their stress at different age groups, and we'll put forward a hypothesis in a minute about why we might be seeing this. So the same version of the gene that's associated with more PTSD is also associated with more rapid suppression of the HPA axis, like I showed you in that earlier slide. So I'm going to shift gears for a second as we talk about other brain regions and how these genetic differences are associated with different brain structure and function. So the hippocampus is one of the areas we know the most about from a perspective of plasticity, but we haven't talked much about this morning. One of the more replicated findings associated with history of childhood trauma, initially it came out with PTSD, then depression, and it looks like the main driver is really childhood trauma, is smaller hippocampal volumes. And Doug Bremner's been one of the leaders of this area. And there's been some controversy as to whether smaller hippocampal volumes are associated with risk for PTSD or outcome of chronic trauma. And it looks like the answer is probably a little bit of both. But one thing then looking at this in our cohort is Nagarfani in our group was able to show that those independent of PTSD status, traumatized subjects with the risk allele had smaller hippocampal volumes, decreased hippocampal activity, and showed decreased integrity of the hippocampal to cingulum bundle in a diffusion tensor imaging study, suggesting that this polymorphism associated with HPA feedback regulation is also associated with some of these classic brain findings that have been associated with PTSD, associated with decreased hippocampal functioning. And again, from the perspective that I first showed, one of the main roles of the hippocampus from the amygdala-centric world is to inhibit the amygdala and help decrease this amygdala output and enhance extinction of fear. But the same version of the gene then is also associated with increased amygdala activation. So decreased hippocampal activation and increased amygdala activation, not putting the brakes on the amygdala. So what about epigenetics? That's in my title. So, I'm just going to give a very, very brief primer. It's the take-home message is the world of epigenetics is changing faster than anybody can keep up with, and it's enormously exciting because we thought with the sequencing of the human genome we'd be done, and then it was just a matter of figuring out the alphabet. And instead, it turns out that as much complexity or more is being done to alter the genome than is already encased in the genome. And so the whole field is very exciting, but there's a lot of work to do. The take-home message from what I'll be talking about today is that there's two main ways of modifying DNA. One is DNA methylation, and the simplistic perspective is more DNA methylation leads to less transcriptional activation. So you just go to turn off that region of the gene. So if you think about the primary sequence being where the genes are, the methylation shuts off per different regions of the gene. And I'm not going to talk about histone acetylation and methylation today, but you can have different ways of unwrapping and wrapping DNA and modulate it in other ways related to that. And multiple stories that others and we have been involved with have shown differential epigenetic regulation or different genes being differentially methylated or switched off related to PTSD or as a history of childhood trauma. And I just want to show you one recent story led by Torsten Klingel and Elizabeth Bender in which the specific FKBP5 pathway I'm showing you is differentially methylated as a function of childhood trauma. So first of all, what they showed you is that, so I showed you the two different gene versions, polymorphisms of FKBP5 associated with high risk for PTSD versus low risk for PTSD in the presence of childhood trauma. What they showed is that that polymorphism that's most key there is in a glucocorticoid response element and leads to differential activity of the glucocorticoid response site. So they showed functionally that that polymorphism alters its expression. What they then showed was using a really cool technique called chromatin capture, was that if you have one version of the gene, the risk version of the gene, you have much tighter binding of the glucocorticoid response element with the transcriptional polymerase that activates transcription compared to the opposite protective version of the gene. And the take-home message is here, the risk version of the gene leads to more glucocorticoid sensitivity or activation of FKBP5 transcription compared to the protective version of the gene. And so that's what's happening at the level of the primary sequence. But at the level of methylation then, again, remember that more methylation leads to the gene being turned off. What they showed is that if they have methylation and trauma, they have if they have the risk gene and trauma, they have less methylation. And this is the way to read these graphs. So they showed this throughout the gene, but primarily in these two sites. So the white and the light gray bar are the no trauma groups with both versions of the FKBP5 polymorphism. But the dark gray and black bar are the trauma history groups. And only if you have trauma and the risk allele do you have much less methylation. And again, less methylation is associated with more FKBP5 expression, which is associated with more rapid feedback, which is associated with more GR sensitivity, which is associated with smaller hippocampal volumes and less hippocampal shutdown of amygdala functioning in the presence of stress. So it's coming together in a really interesting way for how these primary sequences may help differentiate individual vulnerability and how that intersects with trauma at the level of the genome. But what is it doing at the level of circuits? Why, again, is it affecting gene pathways with a history of childhood trauma but not adult trauma? And Regina Sullivan did some really interesting work to suggest how this may happen so that you have developmental critical periods that are cortisol sensitive. So what she showed is that the amygdala effectively doesn't turn on in a rat until about day 10 or 11 of age, postnatal. And so they showed this by doing paired odor shock conditioning and then looking at odor only. And they found in adults, if you pair an odor with a shock and then ask if they prefer that odor or a novel odor, they'll spend less time with the odor that was paired with shock because they're now afraid of it. They're avoiding that odor. But if you do this in a young animal up to postnatal day 9, they have the opposite effect. They actually spend more time with the odor that was paired with shock. It's a very interesting effect, and it holds through adulthood and has some interesting ideas about trauma and repeated trauma and these sorts of things being approaching the negative valence stimulus. But from a most simple teleological perspective, the idea is before about P9 or P10, they are essentially entirely dependent on their mom. Any salient being stepped on or anything else, they need to go towards the mom to get the milk. After that, they start walking away from the nest, they start opening their eyes, they start better learn to be afraid of the world. And so that the amygdala may not turn on fear until after you're past this early, very dependent stage. But what they showed is at this very early age, they could shift the amygdala to turn on earlier during development with a burst of corticosterone. So that if they gave cort, the animal at this P9 age acted like a later older animal or an adult animal. So they could shift the amygdala to be afraid later, to be afraid like an adult earlier during development. So what does that have to do with what we're talking about today? Well, in honor of Bruce McEwen, who has an injury and can't be here, I want to show this paper by Sonia Lupin and him, in which they showed that simply mother's socioeconomic status, which is our population, is associated with much higher levels of salivary port in the kids of those moms compared to not. So we raise a question of, if you have a combination of low socioeconomic status, high environmental stress, and then a subpopulation that then has high child abuse or childhood maltreatment, you have high court at earlier developmental critical periods. So that the role of the amygdala as you grow to combine the external world with internal memories and separate out this hardwired stress response versus being resilient or tolerant and being modulated by hippocampal and prefrontal areas, that's all shifted during this critical period by court levels. So that early life stress through cortisol, through dopamine, through these other processes is activating the brain to be more stressed and fearful by turning the amygdala on. And that's being modulated then at these critical periods by these court responsive genes that are involved in cortisol sensitivity, cortisol regulation, and top-down CRH regulation. So that it suggests that child abuse combined with biological risk factors may lead in a specific at-risk population that has the misfortune of the risk alleles plus the abuse to have an amygdala-dependent emotional circuitry that's always primed for stress responsiveness. And thus, when an adult trauma occurs, they're more at risk for PTSD. So this is a hypothesis, of course, and there's more answers than there are questions, but it leads us to what we think are at least some very specific hypotheses and questions we can ask. So in my last few minutes, then I want to shift gears and talk about the idea of, could one understand intergenerational trauma? And there's been a lot about this over the years. Rachel Yehuda's done a lot with Holocaust victims, and we know that offspring of Holocaust victims are at higher risk for PTSD themselves. There's metabolic stories such that survivors of the Dutch hunger winter and the Swedish famine, that multiple generations in, they have altered metabolic syndromes. But it's been hard to study. And even if you believe the phenomena, there's many, many reasons why it may happen. It could be through differential parenting, and a lot of what Michael Meaney's work has been focused on is the parenting component. It could be through observational learning. A PTSD or depressed mom interacts with the kid, as per all of the things we talked about with the developing child at Harvard, in a different way than the mom who's not. It could be the shared risk environment. Our kids are in an equally traumatic environment as their parents were. It could also be shared genetics. They inherit these risk genes I just talked about. But to what extent is there an epigenetic component? And our own data in the humans is relatively limited on this, but one interesting finding is that if we take moms, so this is in our smaller population, we've interviewed about 150 kids now and done physiology with them, that kids startle in the dark. So you turn the lights on, you play a startle sound, they jump more. That's, if you leave all other variables out, that's entirely predicted by mom's history of self-reported abuse. So mom's history of abuse, moms from a high abuse situation, their kids independently are startling at much higher levels in the dark. Just one of many, many kinds of examples. So how can one study this question? There's been a number of recent studies that are quite interesting. A Nature Neuroscience paper out by Isabel Manzui just in the last few weeks on sperm microRNAs that regulate transgenerational inheritance. Tracy Bale's done some great work on stress exposure. And Eric Nestler's done some really exciting work on paternal transmission. The problem with all of these, and the problem with generally with these stress models, is it's hard to study stress and it's hard to really know what exactly we're putting our finger on because it affects so many different things. So our question's been, could we study such a phenomenon but in a much more simple, reduced model to say, could we understand, basically narrow down a very simple model of intergenerational transmission to understand its mechanisms? And I'll give you a brief history, exposure to the olfactory system, which is where we landed. So this is my background, which is part of why we thought this would be a good area to go. The take-home message is that in the nose, there are millions of neurons, the olfactory sensory neurons. Each neuron expresses one type of odorant receptor only out of about 1,000 different odorant receptors. And then they project through the cribriform plate into the olfactory bulb, and all the neurons expressing the same receptor project to the same glomerulus. So this was work initially identified by Linda Buck and Richard Axel, and Peter Momberts in Richard Axel's lab made a mouse that had a reporter gene, a LAC-Z gene, that was driven only by one of these kinds of odorant receptors, so that you could follow the axons into the bulb of the mouse and see the glomerulus in those bulbs. Now what was cool, and we found by Seth Jones several years ago, is that this process is plastic. If we do this and we train the animals to be afraid of the odor that activates this gene in the adult, they get basically a significant growth of neurons in the nose and more axons in the brain innervating that particular odor-sensitive pathway. So that was really neat, and we're doing a lot to try to understand what are the mechanisms of this adult-mediated plasticity. But Brian Diaz in the lab went one step further. He says, what if we train these guys and then mate them? And this is what the data look like. If you now take the F1, so we're taking the first generation offspring of either dads who were just home cage, dads who were trained to a different odorant, propanol, that doesn't activate this. Those look exactly like home cage. Now I'm going to show you here the offspring of the fathers that were trained to this activating odorant, and the offspring were entirely naive their whole life. They inherited this structural representation as if they had been trained to be afraid of this odor. And Brian showed this 8 ways from Sunday quantitatively. He also showed that if he then takes these F1 generation naive mice, lets them grow up, mates them, they've never smelled the odorant in their life, they have second generation offspring. Those second generation offspring maintain the enhanced representation of that olfactory pathway. Furthermore, he showed that if you did it in females, you could also have the effect and cross-foster the females at day 1, but it went with the biological mother, not the cross-fostered mother. All of this suggesting that the process is inherited and not taught or observed. And behaviorally, he showed, although we only have the marker for the acetophenone gene, he could behaviorally show it goes in both directions as a function of what the father was trained to. So these are actually the grandchildren. And if the grandfather was trained to be afraid of acetophenone, the grandchildren startle more to acetophenone. If the grandfather was trained to be afraid of propanol, the grandchildren startle more to propanol. So it looks like it double dissociates. So it looks like it's inherited. So how do you prove that? Well, what's nice about males is we can take their sperm and we can send them across campus and have them genotyped or make new animals. And what he basically showed that if we took the sperm from these fathers after training, sent them to the transgenic facility, that their offspring from the IVF-derived animals had the same increases in glomerular size, suggesting this is purely mediated by something biological in the sperm. And then the final icing on the cake is he was able to show if we look at the epigenetic, the methylation status of this olfactory receptor in the sperm compared to a different olfactory receptor that is not responsive to acetophenone, we found less methylation in the trained offspring, the trained adult sperm, suggesting that less methylation at the activated receptor in the sperm in the next generation leads to less methylation in their olfactory epithelium and more likely to have a stochastic choice towards that receptor versus a different receptor, leading them to be more sensitive is the current hypothesis. So on a hopeful note, to end, Philomene Morrison in the group has now shown that if you train them to be afraid and then extinguish them to be not afraid anymore, you reverse the phenotype in the adults. And Brian's pilot data looks like if you extinguish them before you mate them, offspring look like they were never trained. So the data are early, but it suggests that whatever's being transmitted is also dynamic at some level. So I've just talked about a lot of things, but the take-home message is that outcome sequelae of childhood trauma, be it PTSD, depression, or other phenotypes, are a function of many different components of the emotional learning process through lifetime. But it's set up by this differential vulnerability, both genetic, epigenetic, and environmental. And I've showed you stories of how the HPA axis can be differentially modulated, as well as potential ways we can study the possibility of intergenerational transmission. So this work has been done by many, many people, and I'm just the cheerleader. The epigenetic work in humans, Elizabeth Bender and Torsten Klingel at the Max Planck, and Philomene Morrison and Brian Diaz, the majority of the olfactory work. Thank you so much for your attention. Yes, Leeway.
Fascinating. So, Kerry, how many generations is this effect can be passed on? And also, I wonder, right? So you can try to extinguish this in F0, but in the subsequent generation, since they never really physically experienced that adversity. Can you do anything about it?
So Liwei's questions were how many generations does it go on, and in the subsequent generations that never actually formally learned it, can we extinguish that? So we don't really know the answer to either of those. The first question is, we know it goes to the second generation, F2. We don't know if it goes beyond that, and are currently working on that, but as you can imagine, it takes a long time to do this. And the second point is we're in process of seeing if we can extinguish those offspring. But in humans, we know, for example, with phobias, even if you have no conscious memory of seeing a snake and being afraid, or being bitten by a dog but being afraid, exposure therapy still works really well for extinction of phobic fears. So we don't think you have to have a conscious memory, or it hasn't necessarily even have to have happened to have the Pavlovian conditioning for exposure-based extinction to work, but we don't know the answer yet. So thanks so much.
Thank you.
Thanks.